Hello friends, today we'll see new two chapters, Amines, which is there for 4 marks with option, and without option for 3 marks. Second chapter is Biomolecules with option 4 mark, without option 3 mark. These two chapters are easy to study, so don't keep these two chapters for options. In physical properties of amines, we see relative basic strength of primary, secondary, tertiary amines, aliphatic amines, aromatic amines. Those things are important, so you go through those things. In chemical reactions of amines, Hofmann elimination reaction, Hofmann's exhaustive alkylation reaction, diazotization reaction, which is also termed as orange dye test, then acylation of amines, distinguish between primary, secondary, tertiary amines using nitrous acid, importantly carbyl amine reaction, carbyl amine reaction. This is a special test for primary amines. Okay, this test is not shown by secondary and tertiary amines. Only primary amines will exhibit carbyl amine reaction. So go through these reaction. Electrophilic substitution reactions of aromatic amines, in that bromination, nitration, sulfonation. Guys, in sulfonation we are going to come across a new term, dipolar ion or Zwitter ion. Many times there was a question for 1 mark, define Zwitter ion. So go through the definition of Zwitter ion 2. In the next half of the chapter, you will see diazonium salts, the reaction pertaining to diazonium salts. In that, how the displacement of diazo group take place using different reagents, namely Sandmeyer's reaction, Gattermann reaction, etc. Even one more reaction is there, that is reaction with fluoroboric acid. Important reaction to prepare fluoro-substituted aromatic compounds. Lastly, coupling reactions. Many times there was a question of 1 mark, define coupling reaction. One should go through this also. Last but not the least, Hinsberg's test, where we are using Hinsberg's reagent. So nitrous acid, very important reaction to distinguish primary, secondary, tertiary amines. So here again the same nitrous acid reaction. Then one more thing I want to tell you here is, usually 2 mark question will be there in exam. Many times they ask this question also, basic nature of amines, explanation regarding basic nature of amines. So 2-3 times we come across this question, so prepare this also, best explanation for basic nature of amines. So this para-toluene sulfonyl chloride, it comes in displacement of azo group, diazo group. See here, simple reactions are there. Hoffman elimination, definition. See again here, basic nature of amines. Defined quaternary ammonium salts, where you will find nitrogen is having plus charge. Simple chemical reactions. Simple conversion reactions. See guys, Hinsberg's reagent. Just few minutes back I have told you about Hinsberg's reagent used in Hinsberg's test. Carbylamine test, popular test for primary amines. This is also called as isocyanide test. Carbylamine test is also called as isocyanide test. See friends, here the same isocyanide test is asked in this way. Effect of this reagent on ethylamine. Ethylamine is a primary amine, and here instead of naming that reaction, they are given in this format. So here one should not get confused with the things. IUPAC nomenclature: lithium aluminum hydride, a popular modern catalyst of reduction. So complete these reactions. So likewise simple reactions are there which needs a lot of practice. Biomolecules is one of the reaction free chapter, or you may say very less number of reactions you are going to find in this chapter. Very less reactions, very less structures you can see in this chapter. Now in this chapter, importantly you have to go through definition of carbohydrate, classification of carbohydrate, monosaccharides, disaccharides, trisaccharides, polysaccharides, etc. After that, preparation of glucose from sucrose, preparation of glucose from starch. Out of these two preparations, one will be there in every exam. After that, structure and properties of glucose. So in the structure and properties of glucose, you are going to see different reactions, very less number of reactions. What happens when glucose reacts with hydroiodic acid? Then what happens when glucose reacts with hydroxylamine, hydrogen cyanide? What happens when glucose undergoes oxidation in presence of bromine water? What happens when glucose undergoes reaction with acetic anhydride? What happens when glucose reacts with nitric acid? It undergoes oxidation. So out of these 4-5 reactions, any one will be there in every exam. So don't leave apart these reactions. Definition of reducing sugars, non-reducing sugars, examples, Haworth ring formula of different carbohydrates, glucose, fructose, sucrose, etc. One should prepare while preparing this chapter. About proteins, definition of proteins, types of proteins, globular proteins, fibrous proteins, their example, structure of proteins, primary, secondary, tertiary structure of proteins, then definition of peptide bond, denaturation of proteins, enzymes, its definitions, functions of enzymes, examples, then thereafter nucleic acid, DNA, RNA, distinguish between nucleotides and nucleosides. Then after that, bases in nucleic acids. You have to practice pyrimidine structures, purine structures, cytosine, uracil, thymine, adenine, guanine. So practice these structures so that if anyone among these is asked, you should be able to draw the structure correctly. See guys, here if you study distinguish between nucleotide side, so you get this answer. There one should able to write the structure of nucleotide and nucleoside also along with difference. Then this is here is the preparation of glucose. Hayward structure, Fisher projection formula is there that you have to draw. Then reducing sugars, non-reducing sugars, their examples, peptide linkage definition, carbon definition, reactions of glucose, preparation of glucose from sucrose, okay, enzymes definition, simple reactions. So whatever I have said initially, those things you are finding here in the previous question papers. See, structure of thymine just at the end of the last slide I told you. Then what is new here in this? See, same questions are repeated in different years. So once you thoroughly prepare these questions, no need to go for any other thing. So 10 years question paper you are studying means you are thoroughly preparing whole chapter. That's what my opinion. So go through these questions properly so that you will get full confidence. Thank you. And finally, like, subscribe and share. Thank you.